Hi, welcome to lesson 6.8. This lesson continues with soil protist symbioses, and we will concentrate more on the very intimate symbiotic relationships, and that includes endosymbiosis. This is when one organism becomes permanently a feature in another organism where they once lived alone, and now they've combined forces to live as a completely new creature. This lesson covers organisms including myxotrophs, those that can photosynthesize and predate on other creatures, microbiotic crusts, conglomerate skin-forming organisms in desert and other extreme soils to protect that soil and to start ecosystems moving forward. We'll cover the wonderful and extreme world of lichens that are little micro ecosystems that join forces and are able to harness extreme superpowers as a result. And lastly, we'll cover protist animal parasites that also live part of their life cycle in the soil, and we'll look at how the parasitic protists in the soil reflect the above ground biodiversity of animals in forest ecosystems. Okay, so let's jump into this very exciting lesson 6.8. So first of all, let's look at myxotrophs. These are organisms that are autotrophic, so that means they are photosynthetic, making their own food from photosynthesis via sunlight. And at the same time, they are heterotrophic, so that means that they require carbon from other sources. So they actually also consume organic matter or other organisms via predation. So basically they have the best of both worlds, where they can photosynthesize and they can also predate. So if there is no sunlight, if they find themselves in a situation where they can no longer photosynthesize, they can survive via predation or through engulfing organic matter, for example. Some amoebae contain green algae, and of course green algae are photosynthetic protists, which we've also described as microalgae. And so these algae are actually symbiotic or endosymbiotic within the amoebae. An example of this is shown here, where there's a testate amoeba and it actually has algae living inside of the amoeba. And sometimes these algae might be consumed and not alive within the amoeba, but a lot of the times they're actually symbiotic and living there and photosynthesizing inside the amoeba. In very nutrient-poor environments called oligotrophic environments, such as marine environments sometimes, where there's not much nutrient available, algae are myxotrophic. So a very nutrient-poor marine environment, algae in that environment are often myxotrophic. Wet sphagnum peat also is nutrient-poor and contains myxotrophic organisms as a result. And an interesting example is myxotrophic flagellate euglena, which contains chloroplasts, so it can photosynthesize on its own without requiring algae, but it consumes hyalosphenia, and this is hyalosphenia pictured here. So it is a myxotrophic testate amoeba containing chlorella microalgae. So there are algae living inside the testate amoeba, and that testate amoeba is consumed by a myxotrophic euglena, which is in itself photosynthetic, having its own chloroplasts, but it can actually hunt these testate amoebae as well. This is how complex these relationships actually get. And these can sometimes occur in soil as well, not just in water bodies. So very interesting relationships develop the closer we look at those microscopic ecosystem interactions. So myxotrophy continues within the protist kingdom, and this is another example, and that of Paramecium bursaria. This is an organism that is very well studied and is a model organism at universities and biology classes to demonstrate that endosymbiotic relationship, which in this case the Paramecium has that with the green algae Zoochlorella. So it's a beautiful organism with lots, up to something around 700 algae can fit into a one Paramecium cell. and they live out their life photosynthesizing, exchanging nutrients, and getting protection from the Paramecium ciliate. And we can tell it's a ciliate because it's covered in tiny little hairs. So it's a beautiful, large organism that is lovely to study cellular biology with, and particularly the concept of endosymbiosis. So the pioneer of endosymbiotic relationships was Lynn Margulis. In 1969, she proposed that symbiosis plays a central role in evolution of early life, that organisms began living symbiotically in order to form more complex life forms. Her manuscript, publishing the findings she has gained from her experiments, it was rejected 15 times before it actually got accepted, and then it was severely opposed, and she was severely attacked by other scientists, mostly men, because women in science did not get a go, and luckily some of them just persevered. But in 1970s, a molecular analysis technology allowed other scientists to find that endosymbiosis is in fact an extremely common way of life for organisms. And in particular, they've discovered that mitochondrial and chloroplast ribosomal RNA is exactly that of bacteria. And so this idea was finally accepted that early life forms engulfed bacteria and allowed those bacteria to serve vital function within the cells. And this is what we call endosymbiosis, when one organism performs a function within another, but it was once a free-living organism and now is no longer free-living but endosymbiotic. So a beautiful relationship, and lots of hardship for some people to present new ideas for the first time. The scientific community can be very, very harsh, particularly if you're a woman back in the 1960s or 70s. So go Lynn. Another extremely important symbiosis is that in microbiotic crusts, where organisms get together and form a layer on the soil surface to protect the soil, and these are conglomerates of several different organisms living symbiotically, and that includes soil protists, particularly microalgae. These crusts in some ecosystems are also described as cryptogamic, cryptobiotic, and microphytic, but all of those do not truly describe the micro nature of these soil crusts. They cover the soil surface to a depth of up to 10 centimeters, particularly in arid, semi-arid, and alpine environments, but really in all environments and across all continents, particularly where there is reduced vegetation cover. They include bacteria, fungi, cyanobacteria, green and brown algae, lichens, mosses, and liverworts. And liverworts are related to mosses, so these are little plants that are non-vascular. And they are critical primary producers. That means that they photosynthesize, containing algae, of course, and cyanobacteria oftentimes as well. And when there is no vegetation cover, they are the primary carbon fixers and therefore primary producers in those environments. They are required for water infiltration and soil and landscape stability in these environments, producing cementing gels and mucilaginous sheaths. And they help to increase plant growth by retaining nutrients and moisture in the environment, and often help seed germination of certain plant species. About 70% of the Australian landscape is arid or semi-arid, and this is also where animals are grazed in native pastures. However, these animals break up these crusts and therefore destabilize the nutrient-rich soil that these crusts provide, particularly around perennial shrubs, helping them to grow. And this significantly reduces vegetation cover in these landscapes. Once animals are excluded, recovery of these microbiotic crusts is slow, and it has been shown that there is minimal lichen recovery after 44 years of excluding animals in Coonamoor, South Australia. The other practice that's engaged with grazing is that of burning woody perennial vegetation to allow pasture growth, to remove the competition for this pasture. And this is quite detrimental to the microbiotic crusts, and annual fires after 7 years have shown complete elimination of the microbiota, significantly reducing infiltration and aggregate formation in that soil, causing erosion and eventual desertification. These ecosystems are actually quite fragile, and to graze them and burn them is going to create desert conditions and will always put the ecosystem back in succession, not allowing them to recover, as nature is always trying to drive ecosystems towards revegetation, and these are the beginnings of revegetation that is being prohibited in about 70% of the Australian landscape. So we do have a problem on our hands there, as these environments need to actually be protected and encouraged to recover in order for us to stabilize our climate, reduce heat, and reinstate rainfall, which requires tree growth. Protists also play an essential role in the formation of lichens. Lichens are actually mini ecosystems of microbial communities, and about 14,000 or 1/5 of fungal species engage this symbiosis by being a mycobiont, and that's the fungal partner that contributes 95% of the biomass of the thallus, and that means the actual entire organism is 95% fungi. The cortex of the lichen is made up of dense hydrophobic hyphal network that has UV protective compounds that are being studied to create sunscreen for us. There's also moisture retention via the cortex and protection from being eaten and other dangers to the lichen. So the microbiont, or the fungal partner in this relationship, is obligate. That means it requires a specific protist algal species or a group of algae to complete its life cycle. So the fungi will partner up with the protist, which is the algal partner, or of related algae, to create the lichen. The interior medulla of the lichen houses the photobiont, and the photobiont is either a green alga or a cyanobacteria, and 85% of lichens actually contain green algae as the photobiont, and this is called the chlorobiont, and the rest form relationships with a cyanobacteria instead, or in addition contain a cyanobacterium as well. So these are photosynthetic bacteria. Most algae can live out their lives solitarily and don't need the microbiome, but are readily lichenized by a variety of fungi or the microbiome. The only restriction the algae undergo once lichenized is that they lose their capability to reproduce sexually and can only undergo asexual reproduction. But in some cases, it allows them to sporulate and even form zoospores that are flagellated. So spores that actually have a flagellum and a motile. So they are restricted to some level, but not entirely in their reproductive capacity. This is an image from the Bunya Mountains that I took on the weekend with gorgeous lichens growing on the tree. And you can see the different species here, the different colors. The lichen is perhaps one of the most famous examples of symbiosis, and it has been known that there are more than one organisms comprising the lichen for over 140 years. But in 2016, Toby Spribil, a scientist who studies lichens, was challenged by a friend to sequence all the lichen organisms. and the DNA analysis was showing that there were many organisms, and he kept thinking that they were just contaminants, that it was DNA that was contaminating the lichen, and yet it turns out that all of these organisms actually are part of the lichen. So there are actually other microbiomes, and these include yeasts, endohyphal bacteria, bacteria that live inside the hyphae of the fungal partner or the microbiome. There are also bacteria that are not endohyphal, viruses, diatoms. There are free-living algae as well, and cyanobacteria. And there are also epiphytic algae, so that means algae that grow on top of the lichen, as well as liverworts. And these are moss-like plants. So each lichen has a different suite of these different organisms, and not all of them exist within a particular lichen. But one lichen called Peltigera hymenina hosts 324 different phyla, and that means different organisms. That means 74 viruses, 236 bacteria, 6 archaea, so these are organisms related to bacteria, and 8 eukaryotes. So those are cells that are non-bacterial, so like, for example, fungi and algae. There are also lichenicolous lichens, and these are lichens that actually live on top of a lichen. So lichens that live on lichens. There are many free-living algae and fungi that lichenize when grown together. So that means they are happy to be separate, but when grown next to each other, they can't keep away from one another, and they will form a lichen. And it might just be a loose symbiosis where it doesn't look exactly like a proper lichen that grows on the different surfaces in nature. They will form some sort of a structure together that is different to how they look when they grow on their own. But each of these partners must do what the other cannot. So basically, they will lichenize if there is a benefit to being in a lichen partnership. But lichens can be thought of as stable relationship networks where life alone is less efficient versus when they get together. They can also be thought of as composite life forms that actually provide entirely new possibilities, and we will see just what is possible when these organisms get together. Merlin Sheldrake, in his book Entangled Life: How Fungi Make Our Worlds, Change Our Minds and Shape Our Futures, which I highly recommend reading, says that lichens are places where an organism unravels into an ecosystem and where an ecosystem congeals into an organism. That's a really beautiful way to describe a lichen, and I really recommend reading Entangled Life. The whole book is full of Merlin's beautiful descriptions of how he perceives the natural world. Lichens play an essential role in ecosystems, and one of their primary roles is that in rock weathering. That means they can literally create soil from rock, having a fungal microbiome. They are very good at mineralizing rock, extracting nutrients directly from rock, and many other substances as well, such as metal, for example. Through their growth into the rock, they produce mechanical forces that first of all breaks the rock up, and then through the digestive process where the fungal hyphae excrete acids and also bind mineral compounds. This means that there is mining or extraction of minerals from that rock. And then as the lichen ages and sloughs off and dies, it produces organic matter. And that becomes the first soil layer where we have the mineral component and the organic matter of the soil beginning to form. Other ecosystem functions of lichens are plenty and varied. They grow anywhere from the Antarctic throughout deserts and all the way into the tropics, living on plants, and some are as old as 9,000 years. The desert crust lichens, so the microbiotic crusts we've spoken about earlier, display succession. So that means that really hardy ones will grow to start with that can deal with very harsh desert conditions. and they then pave the way for other lichens, bringing in more complex symbiotic relationships. And of course, they serve to increase soil stability and act as a nutrient sponge and really kickstart that ecosystem succession. Many can withstand drought through dormancy of around 100 years. And the way that they colonize ecosystems is that fragments of their entire lichen can move to other areas via animals, by wind. As they break off, they actually are able to travel. And they are thought to be microbial reservoirs that seed barren habitats with crucial bacterial strains. Because they harbor bacteria as part of the lichen, they can actually carry that bacteria across to start the process of ecosystem formation in barren habitats. The microbiont, or the fungal partner, is able to reproduce sexually and form spores. So those spores can be dispersed by wind or animals. And once the spores germinate, the fungi needs to then find a photobiont partner, so an alga or a cyanobacteria, in order to become a lichen and complete its life cycle. In forests, lichens protect young trees from herbivory because they create lots of biochemicals, compounds that protect the lichen and will also protect the tree as a result. They're also bioindicators of climate, air quality, acid rain levels, and spatio-temporal habitat continuity. So we can study habitats over space in time by looking at the lichen community within that ecosystem. Coastal lichens are able to capture nutrients from ocean air, and by doing so, they leach those nutrients to the soil layer. So they capture nutrients from the atmosphere and direct them down to the soil, enriching that soil ecosystem. And also, as they decompose, they will release those nutrients into the soil. In temperate rainforests, up to 50% of the nitrogen is thanks to the cyanobacteria in the lichen. And their surface topography, as you can see here, for example, is quite intricate, and it is able to create humidity and maintain a certain temperature. And this supports other epiphytes, so other organisms such as orchids, for example. It provides benefits to those plants that grow as epiphytes by regulating humidity and temperature. Other native plants also benefit, as well as arthropod and microfauna communities. All these organisms take advantage of the lichen, and the animals can actually feed on lichens as well. Lichens are also critical winter forage for larger animals in temperate and boreal forests. Elk and other larger animals that are herbivorous rely on lichens when plants can't photosynthesize during winter months. So they do provide a lot of incredible benefits to ecosystems and are an integral part of those habitats. For a long time now, scientists are recognizing that lichens are very extraordinary. They have quite extreme superpowers. They create over 1,000 unique secondary metabolites that allows them to survive acidic air pollution, confer heavy metal tolerance to lichens, and they can withstand many other pressures, such as extreme UV exposure, for example. So they can be thought of as extremophiles. So these are organisms that can survive in extreme conditions. And because there are several organisms in the lichen, they can be viewed as polyextremophiles. So for example, they can be frozen in liquid nitrogen at minus 195 degrees Celsius and be thawed out and still live. They can even survive outer space and the conditions on Mars, and withstand 12,000 times the human lethal dose of gamma irradiation that is present in space. So this interests astrobiologists because they are interested in this theory of panspermia. And this is where some organisms can be catapulted from Earth via meteorite, for example, hitting the Earth's surface, and life forms being transported on rocks that are catapulted out into space and starting life in space on other planets. Is the same possible in the reverse? So astrobiology is studying whether life on Earth has been seeded via organisms coming from outer space and starting life on Earth. Lichens can also withstand up to 50 gigapascals of pressure. And to give you an idea, that's about 100 to 500 times greater pressure than is present at the deepest place of Earth, which is the Mariana Trench. This kind of pressure is equivalent to a meteorite dislodging a rock with enough pressure to escape the velocity from Mars. So that is quite extraordinary, and lichens are being studied extensively for their extremophilic or extreme liking capabilities, and they can only do that when they're in the lichen symbiosis and not when they live singularly as the individual organisms. And this is a photo from the Bunya Mountains of a lichen growing on rock. Different kinds of lichens. You can see all the little colonies there. And an extreme example of a lichen I found was on the roof when it was around 40 degrees Celsius ambient temperature. And on that roof, it must have been way above that because it's a metal roof. And there's a lichen just growing on that roof. Extreme conditions, drought, heat, and that just boggles the mind. Lichens do not look anything like the organisms that form them, and they form very beautiful shapes. This one was found at the Bunya Mountains, must have fallen high from a treetop, but this particular branch actually has many different kinds of lichens in it, and that's why that photo is a good representative of the different forms that different lichens form. Some are crustose, and so they have this crusty appearance. Some are foliose, look like leaves, such as this one. Some are squamulose, so that means they look scaly. Some are leprose, which means that they look dusty. And some are fruticose, and that means that they are branched. And this is probably an example of a fruticose lichen. So just to show you some of the examples that I have found at home and in the Bunya Mountains, there's one growing on the driveway, just on a decomposed granite surface, forming quite a large colony, so forming soil as it grows. This is one from the Bunya Mountains, a beautiful red lichen. It must have red algae in it. I'm not sure what gives it this beautiful color. And of course, on trees, they form different beautiful shapes, oftentimes looking like little maps on trees and rocks. This is probably a foliose and fruticose lichen here on a branched one. Possibly, I'm not really exactly sure how to place them in those categories. And there's another fruticose one with beautiful branching style of growth. And the little maps formed on a tree. And you can see the little demarcations where the little colonies mark their boundaries with various chemicals to claim their space on the map. So that's it for lichens. This is the end of the story of the lichens. And the last story is that of the soil protists that parasitize animals and are therefore a great indicator of the ecological biodiversity above ground. So that means that many soil protists can be found in soils as animals and invertebrates that contain these protist parasites shed them into the surrounding soil. Apicomplexa is a group of protists that are parasitic on animals and invertebrates, and in soils of tropical forests of Costa Rica and Panama, Apicomplexa is found in abundance, and there's a large diversity of protists that are actual parasites of animals and invertebrates. So that means that we can actually monitor entire forest ecology via these parasites being found in soil. So if we know that a particular animal is associated with a particular protist parasite, if that parasite is in the soil, it indicates that the animal which it parasitizes must also exist. In alpine soils, parasite and animal diversity is also correlated when examining litter and mosses of that alpine soil environment. So protists in soil are a good indicator of what is happening in the entire ecosystems when these protists are also animal parasites. So we've come to the end of lesson 6.8, And in our next lesson, lesson 6.9, we'll look at soil protist ecology and investigate a little bit how protists affect plants, so the protist-plant interactions. We'll also cover some other topics related to the role of soil protists in soil restoration. So you'll be hearing from me again in lesson 6.9. See you then.